Hello and welcome to the Investigating Transpiration revision video from scienceworksheets.co.uk. Please like, subscribe and comment to support the channel. Transpiration is defined as the loss of water vapour from the leaves of a plant through the open stomata. A potometer measures the rate of water uptake by a shoot. The potometer does not measure water loss from a shoot. However, it's assumed that the rate of water uptake is almost equal to the rate of water loss by transpiration, because most of the water taken up is lost through the leaves. Some of the water taken in by a plant is used for photosynthesis and some is used to keep the cells turgid. This is how to use a potometer. You will prepare the potometer by filling with water and removing all the air bubbles. Submerge the shoot in water and cut a fresh 2 to 3 cm section off of the stem underwater. This is to prevent air entering the xylem. The stem should be cut at a slant to increase the surface area. Once you have cut the shoot, immediately attach the shoot to the potometer underwater or in a water film and seal with rubber tubing or grease so the connection is airtight. Place the potometer vertically in a clamp. Introduce a single air bubble into the capillary tube if using a bubble potometer. This is done by lifting the capillary tube out of the beaker of water for 1 second, then placing it back into the water. Record the bubble's starting position in millimeters on the ruler. Set up your environmental conditions such as a fan for air movement, which blows onto the leaves. Now start the stopwatch and after 60 minutes stop the stopwatch. At the end of 60 minutes record the position of the bubble in millimeters on the scale. Do at least 3 repeats with different shoots. Between repeats, reset the bubble to a known starting position. This is achieved by opening the reservoir tap. We need to be able to change the environmental conditions that the shoot is exposed to, so we can see how this affects transpiration. So to alter light intensity, the lamp is used at different distances from the shoot. For air movement or wind speed, a fan is used at different power settings, low, medium or high. Alternatively, for air movement, we can use the same power setting on the fan, but move the fan different distances from the shoot. For humidity, we can increase humidity by placing the shoot and potometer inside a plastic bag or bell jar with some damp cotton wool inside. This will increase the water vapour in the air around the leaves. To decrease the humidity, we can use a dehumidifier. For temperature, to increase the temperature, we can use a heater, or to decrease the temperature, we can carry out the experiment in a different room that is colder. Calculating the rate of transpiration. The rate of transpiration is the change in volume, or the change in the distance moved by the bubble, divided by the time taken. So if the air bubble in the potometer moves 25 millimeters in 5 minutes, then the rate is 25 divided by 5, which is 5 millimeters per minute. There is data analysis work which is needed for this topic. This is in our revision notes at our website. See the link in the description. Thank you for watching.